All right, I think we should get started. Good afternoon, everybody, and welcome to Hydroepidemiology. This is the session that we are organizing for like last 3 years, and it's an attempt to understand how hydrology can play a role in betterment of human health. So we have exciting session today. Unfortunately, few of our invited speakers couldn't be here, so but we have their representatives who will talk on their behalf. So our first speaker today is was actually it was Dr. Andrea Rinaldo, and it was an invited talk, but he couldn't be here, and Lorenzo Rigotto will speak on his behalf. And so the way we will do it is like we will have 12 minutes for talk and then 2 to 3 minutes for questions. And in the meanwhile, I'll request the next speaker to be ready with their talk. Thank you. Lorenzo?
Thank you, Dr. Pratt. And first thing I would like to convey Professor Rinaldo's apologies and regret for not being able to be here today to present. So the topic of this talk is the definition of a threshold for epidemic onset in spatially explicit models of waterborne diseases. So as a general motivation, maybe some don't expect waterborne diseases to be one of the major causes of death in the world, especially among children and in developing countries where health systems and sanitation measures are poorly implemented. Our aim as modelers is to build process-based models that can help in predict the outcome of outbreaks and possibly plan intervention measures and strategies to mitigate them. In this talk, we try to define conditions for the spatial onset of diseases. So in classical epidemiology, this condition is already defined as the basic reproduction number, which is practically defined as the number of secondary infections which are caused by a single infected individual introduced in a new community. What mathematical epidemiology does in standard models is to consider human communities as local, independent systems, where they usually keep track of susceptible individuals, healthy ones, infected people, and also, this is a feature of waterborne diseases, the concentration of pathogens in the aquatic habitat. This is because infection is caused by ingestion of contaminated water. So this compartmental model can be expressed through these equations, where one can appreciate demographic terms, infection functions depending on the concentration of vibrios in the local system, some mortality of infected and their recovery rates, while in standard expressions the reservoir of vibrios in the environment is replenished by human excretion but would normally die in the absence of it. So this system has two equilibria. One shows no infectives, so it's called disease-free equilibrium, and the other converges to, in the other, this system converges to a positive number of infectives and bacteria. So one can find a threshold in here because these two equilibria are alternatively stable. So for any initial condition, depending on the parameter setting, the system will converge either to one or to the other. For those who don't know, a stable equilibrium has a Jacobian with which has eigenvalues with negative real parts. So the condition one can set is that one of these eigenvalues changes sign, and this is how basic reproduction numbers are found in this framework. So as a combination of parameters that causes a change of sign in the eigenvalue and causes the exchange of stability from the disease-free equilibrium to the endemic one. So you can see here it's positively connected to the infection and contamination rates, and on the opposite side, negatively correlated with infected and Vibrio mortality. So this is what happens when R0 is greater than 1. So you have convergence to a coexistence, let's say, of susceptible and infected. And this is what happens when R0 is lower than 1 at local scale. So an exponential convergence to 0 infectives. What our lab's work has been in these years is to extend this framework, local framework, to a network model where we have a number of human communities, each one described by this kind of model, but with some terms describing the transport between each of the nodes. So hydrologic transport of Vibrios, but also human mobility patterns on another graph, which is completely connected, usually. So in this case, we have a system, n systems of these ODEs, where n is the number of nodes. And you can see here new terms appearing, some describing the transport of Vibrios on the hydrologic network, other ones describing the human mobility, like this. Usually we don't describe a human mobility as actual movement of people, but we keep track of the people getting infected, susceptible people getting infected in other nodes while traveling, and the contribution of moving infected people to node I in the human excretion term. So we applied the very same framework that we have seen for the local model to this more complex system. The concept remains the same, as there are still two equilibria, one disease-free and one endemic. One can show that the bifurcation condition that I was talking about equals this condition, that the determinant of the Jacobian of the linearized system around the equilibrium equals 0. We make use here, after complex calculation, which I don't show, of this matrix G0, which contains information about the hydrologic connectivity, which is this P matrix, plus a term which contains information about both the parameters of the system, this P beta, but also about the structure of the human mobility fluxes, this Q matrix. So one can see that the possibility for the disease to cause an outbreak in the system is not only dependent on the local parameters, but also on the structure of human movements and of the morphology of the network. So one can prove also that this the outbreak can start only if the dominant eigenvalue of G0 is greater than 1. So as a first result that we obtain on theoretical network, we show here that in this space of the parameters B and L, which describe the movement of particles in the hydrologic network, we show here that the line corresponds to a constant basic reproduction number, so local R0. So to the left of the lines, the epidemics can start, while on the opposite side, they cannot. So there are cases in which the epidemics can start for values of local R0 lower than 1, and vice versa, cases in which it cannot start for R0 greater than 1. For instance, for high values of L, which defines the residence time of particles in a node, you need a higher R0 to sustain the epidemics, as L defines also a flushing out of the system as particles are driven downstream. So for more, our application studies, we also would like to test this framework as a predictive tool. To this aim, we look at the dominant eigenvector of G0. So the eigenvector defines the directions the system is evolving to around the bifurcation. So around the bifurcation, the disease-free equilibrium loses stability and is a saddle That means that in the state space it has two attracting directions, two diverging ones. So we are on the diverging direction, and we look at the infective components of the eigenvector, and we project it on the map to see how well they comply with the actual observed cases. And actually we find in this theoretical example that the components of the dominant eigenvector have a very good correlation with the outcome of the outbreak, even though there is no clear population pattern that could drive the disease. So as a first application we tried, we used the model I showed before, which we calibrated for the Haitian epidemic from the start to May 2011. And we compared the values of the dominant eigenvector to the observed cases at the time of the peak, to obtain quite a good correlation, which was around an R-squared of 0.9. And what we observe also is that if you want to avoid the outbreak of the disease in the country, you need to operate on the beta and P parameters, that is on the contamination infection rates via vaccination or sanitation. Rather than mobility, it's more complicated to change the outcome of the disease operating on mobility parameters. So this was an ex post evaluation. So to further test the predictive power of this tool, we ran this experiment on the Tukela in South Africa case, where an outbreak was present at the beginning of the last decade. So we calibrated the model on just a few weeks of the epidemics between these two lines, and we compared the values of the eigenvector components at each node to panel C, to the actual observed cases at the time of the peak, panel B. So even though the correlation might not seem striking, the R-squared of the two is around 0.9 also here, and it's quite robust to parameter uncertainty, which you may expect to be high when you calibrate on such a limited data set. So in general, we conclude that R0 can be a dangerous, let's say, threshold or indication of the potential spreading of diseases in a system. And one has to take into account the more complex interactions of different communities. The robustness also of the predictions of the dominant eigenvector proves that this tool can be really used for assessment of intervention strategies, which is really a key goal of our work. So here, I'd like to thank all the group which participated in this project. Thank you.
All right, we have time for 2-3 questions. Yes. We don't have mic.
No, we don't.
Can
you repeat the question?
Yes, please.
Well, this is an interesting question.
Repeat the question.
Okay. So the question is, if whether we have tried to test the model in an area with no cases present. So this presents some difficulties as all the application studies are made with the same model but with specific tweakings. But in general, I think one of the most interesting applications would be to really define the proneness of a region to epidemic onset based on the structure of the network and population distribution. So an a priori evaluation of the risk that also a number of hypotheses can be tested, also distributed parameters, which we have done for South Africa. So really to see how the local conditions can control the epidemics. So you can also try to understand spatial interventions, so locally based, and but still we haven't done anything. Yeah.
All right. I think we have to move on to a next speaker. Apologies, but you guys can discuss later on. Okay. Dr. Pascal has some questions for you, but you can later. Yeah. Yes. All right. Next up, next speaker is Teresa Yamana. Please.
So I'm going to talk about some recent work we've been doing in trying to relate malaria and climate change in West Africa. So the questions we're looking at is, can we identify some regions within West Africa that might be more sensitive than others to changes in malaria transmission as a result of climate change? And then we're going to look at a way about how we can try to start to quantify some of these changes. So malaria is very closely tied to climate. The primary vector of malaria in West Africa is the Anopheles gambiae. This mosquito breeds in temporary puddles of water formed by rainfall runoff. So the mosquito needs these rainfall puddles to breed in. There's also temperature dependencies. The mosquito has an aquatic stage. These development rates are dependent on temperature. And the lifespan of the adult mosquito is also a function of temperature. There's also a lag time between when the mosquito acquires a parasite and when it becomes infectious. And this is also a function of temperature. So we can summarize some of these climate dependencies in this quantity we call the vectorial capacity. So this is defined as the number of infectious bites that would happen as a result of a single infectious person per day. So all of the mosquitoes biting this one infectious person and then distributing those bites onto subsequent people. So this is a function of the number of mosquitoes, how often the mosquitoes bite. These are functions of both temperature and rainfall. And also some of these temperature-dependent rates, so the probability that the mosquito lives long enough to become infectious. So thinking about malaria transmission and climate change, of course, we're less interested in what happens to the mosquito than we are to what happens to the people. So the prevalence, this is the proportion of people that are infected at a given time. We can think of the change in prevalence when climate changes as a combination of two components. The first is how will the prevalence change as a function of as vectorial capacity changes, and then how will vectorial capacity change in response to the climate. So here we're trying to estimate these two quantities. So prevalence data in this region, as many other types of data in this region, are very sparse. Here we have a map of estimated malaria prevalence produced by the Malaria Atlas Project. So the gray regions are areas that are close to no transmission, so less than one infected person per 10,000. And then the colored regions are areas where we see stable or expect stable malaria. So now we want to define another rate. This is the entomological inoculation rate. This is the number of infectious bites that a person in an endemic region might receive in an entire year. So at very low levels of the EIR, small changes in the EIR have a big change in prevalence. So if you're going from one infectious bite a year to 0.1, this is a big difference. Whereas at high levels of EIR, changes aren't as important, because if you're already getting 100 infectious bites a year, your additional bites aren't that important because you're probably already infected. So it's a very nonlinear relationship. So Smith and some other people at the Malaria Atlas Project have used a compartmental model, sort of like in the previous presentation, to find equilibrium values. And in this way, they were able to relate the EIR to the prevalence. And they also have an equation for vectorial capacity as a function of the EIR and the prevalence at steady state conditions. So because what we want to know is the relationship between prevalence and vectorial capacity, we've rearranged these equations that they found to find vectorial capacity as a function of prevalence. And so this is shown in these two graphs, so on a regular scale, and then in the bottom, this is the log scale. And basically, we see a similar thing as we do in the EIR. At small levels, there's more sensitivity to changes in vectorial capacity than there are at higher transmission rates. A key difference is this critical value of vectorial capacity, below which transmission can't occur. So you don't get steady transmission at these low levels of vectorial capacity. And the exact value isn't quite known. These are functions of the parameters of the model. So the duration of the disease infection and the transmission efficiency. So if we then take this relationship and apply it to the prevalence estimate, we can estimate vectorial capacity in this region. So going from about 0.01 in the north, and the highest is around 4. And the gray regions, again, these are the areas that had the very low prevalence. So we can think of these as regions where the vectorial capacity is below that critical value. And this makes sense because this is going into the Sahara Desert where it's very hot and dry, so hostile conditions for mosquitoes. Okay, so really what we want to know is how the prevalence will change when the vectorial capacity changes. So we numerically differentiate this relationship that we found. And so this bottom graph is the change in prevalence for percent change in vectorial capacity. So we see it kind of maximizes right as it approaches the critical value, and then it drops down to 0. And because we don't know the exact value of that critical value, I've continued the curve in red. So then if we take this curve and we split it into 3 equal regions, we get an area of highest, medium, and lowest sensitivity to changes in vectorial capacity. Now we can put these on our map of vectorial capacity and outline regions that we think are highest, have the highest sensitivity to changes in vectorial capacity, medium, and lowest. And again, those regions where we weren't sure what the exact value was because we didn't have the prevalence data, we know that most of these areas are very low, so that's going to be in the low transmission area and maybe a small band of medium sensitivity. So we don't know exactly where those boundaries are. Okay, so once we have the relationship between the prevalence and the vectorial capacity, so now we want to estimate how vectorial capacity is going to change in the future climate. Okay, so this is our map again, and we've chosen 4 locations, 2 in the high sensitivity region, 2 in the low sensitivity region, or the medium sensitivity region, and we'll call these 1, 2, 3, and 4 from north to south. Right, so this is our same graph, so the four locations there. So to find the vectorial capacity at these climates, we need to use our malaria transmission model. So this is HydroMats. This is a high-resolution model of hydrology and malaria transmission. So 2 main components. The hydrology component runs at the village scale. It's 10-meter resolution at an hourly time step. So this takes inputs of temperature, rainfall, all the environmental data, and it simulates the water pools. So it simulates overland flow, infiltration, evaporation. So for every time step, we have an output of the locations of the water pools, as well as their depths, their temperatures. So these provide the input for the mosquito component. So the mosquito model is an agent-based model. We have individual mosquitoes that lay eggs. The eggs, assuming that the pool lasts long enough, the eggs will go through their development cycle, emerge as adults. Again, that's temperature dependent. The adults will then fly around, look for the village, find somebody to bite, and then they'll take their bite, lay eggs, and so on, and they can transmit the disease. So to find the change in vectorial capacity, we model under future and current conditions. So this is, in the dark bars, we have the current conditions. So hot and dry in the north and going cooler and wetter as you travel south. And I want to mention that this is a region where the climate predictions are very uncertain. So of the current climate models, about half of them predict a wetter climate and half predict a drier climate. So to illustrate our point here, we selected the most extreme example, so the wettest prediction. Not to say that we think that's the most accurate, but we want to see maybe what's the biggest change in vectorial capacity we can get. So those changes are shown in the lighter bars by the end of this century. Okay, so we model under the future and the current climate, and then we look at the change in vectorial capacity. So in the top and the bottom regions, the highest and the fourth region, we get increases in vectorial capacity, and we actually simulated a significant decrease in vectorial capacity in the other two regions. So if we combine these two components then, we have the change in prevalence due to the change in vectorial capacity in the first column, and in the second column, the change in vectorial capacity as a result of climate change. We can kind of look at how these two will combine and multiply them to get the change in prevalence with the change in climate. And applying those to our current prevalence, we can then get the future prevalence, and that's what's shown in the bar graph. So again, because we chose that very extreme value of a very wet future climate, we're less interested in the exact numbers here. But a few things we want to observe. Well, this method lets us look at the two components and look at how they compare with each other, which ones are bigger, which ones are having a bigger effect on the change in prevalence. So some of the things that stand out, the main thing we notice is that the change in vectorial capacity due to climate is actually there's more variance in that between locations, whereas there's less of a difference between locations in the sensitivity. But that's not to say that this difference in sensitivity isn't important, especially if we look at our fourth location. We had a nearly doubling of the vectorial capacity, but because this happened where the sensitivity to the change in vectorial capacity wasn't as high, the overall change in prevalence was less than it might have been if we, for example, had that same change in vectorial capacity in the highest sensitivity location. So this is just kind of a way where we can look at the different components, compare them, see how they're going to play and shape the future of malaria transmission. So in conclusion, we identified a region that we expect to have the highest sensitivity. So areas that are very low, transmission will probably continue to be very low because even an order of magnitude of increase in vectorial capacity might not be enough to sustain transmission. But then there's this middle region which is more sensitive to changes, and then areas that already have very high transmission are also less sensitive. So this area happened to be along that northern boundary of current conditions. And then there's the two components of the areas that are most sensitive to changes in prevalence and areas where the vectorial capacity is supposed to change the most. And so these two regions don't necessarily overlap. So we have to look at what happens in each of these components and how they'll interact. So,
Great.
Thank you.
All right, we have time for 2 questions. Yes.
It's my understanding that the malaria mosquitoes need actually water pools to maintain their life cycle. So when you were looking at changes in precipitation, did you model or get data that suggested how the temporal changes in precipitation were going to occur? Because I would think that would be as important or more so than just the total amount if in fact the rain was distributed in a different manner so that the pools did not maintain themselves or more pools were generated than had been in the past.
Yeah, so the way we projected the change in rainfall, we used a hydrology model to translate the rainfall into the changes in pools. So for current conditions, that was pretty simple. We fed in hourly rainfall data, and through the overland flow process, we actually got an hourly distribution of these water pools and how long they last. So to predict the future conditions, we actually assumed that the future wetter climate would take the form of a shift of the climate. So we looked at where rainfall is currently, the levels are currently as high as they're projected to become. So we moved kind of up the climate gradient. So we use the temporal distribution of a current rainfall in an area that rains more than it is currently. So we're making the assumption that the future rainfall will take the same pattern as we're seeing in a wetter region under current conditions. So that's an assumption that we've made.
All right. One more question. Yes, yes.
It's not a related question, but for the other term, so you have the relationship between prevalence and vectorial capacity in a sort of equilibrium situation. But as you get to some of those low transmission regions, you have extremely seasonal dynamics, in which I wonder whether that overall relationship and its derivative may be the way to look at.
Yeah, that's an interesting point. So that definitely the relationship that we calculated using that equation is it's just one number for the location for the entire year. Whereas with our model, we actually get this hourly time step of the vectorial capacity, and we do see much bigger peaks, of course. It's close to zero for most of the year, and then in the rainy season, it's very high. So I think one of the ways we can get around that is move from this very simple equilibrium model to a more sophisticated model. Our model, we're working on an immunity model so that we can actually have an agent, actually show the mosquitoes transmitting the disease on a much more fine resolution level. So I think there's some medium between this very simple equilibrium model and a very, complex model.
All right, we need to move on now. Thank you so much, Teresa. Our next speaker is Martina Merwe, and she's going to talk about cholera in Africa.
Forward and backwards. Sometimes it's easier to look over here and squiggle it around. I don't know which way.
Good afternoon. I'm Marna van der Merwe from South Africa. I work for the Council for Scientific and Industrial Research. And my colleague, Dr. Matala Dupree, and I have been involved with cholera research in the region for the last couple of years. And today's talk will be about developing or trying to develop an explanatory prediction model for cholera outbreaks in Africa using rainfall patterns. So the talk is just structured around what is the issue, why did we bring this up as a problem statement, and I just want to give you some background. I'm sure a couple of you have worked in Africa and are aware of the situation of cholera in Africa, but just in terms of the cholera incidence and rainfall patterns in Africa, the approach that we implemented to try and develop such a prediction model, some results, and a short discussion on the conclusions and acknowledgements who have been involved with this type of work. So the problem statement came around from our work that we did in Mozambique, in Beira. It's a harbor city, where if you ask the locals and say, do you know when cholera will occur? And they will tell you, yes, when the first rains start, we know cholera will appear. And we thought, but that's an easy way to go around it. We'll just develop a correlation model. And we developed a model that we calibrated against the cases. But the idea was just to use rainfall in the end. And because we could get a rainfall with the rain 2 weeks after a threshold was reached, then the cholera cases would start. This model could explain close to 75% of the variability in the cholera cases. But the downfall was 2001 was an exceptional year. The rest of South Africa reported their biggest cholera outbreak to date. Beira barely had any cases. The model tried to predict those cases, and it was totally outside of what was reported. And 2002-03 also showed, but not as extensively, a similar problem. So, possible reasons for what we actually experienced there, we attributed to quality of data, because the health data is not very, it has got a lot of gaps in it over space and time. It's overestimates, underestimates. You do not know what you can use or not use. So you try to actually model with this data, try to explain correlations, but actually you cannot interpret these correlations because you do not know how good the data is. Also the methods that you implement in terms of gap filling or validating data, or which data sets to use, we found was quite an issue. And then, of course, underlying assumptions regarding the data where you actually miss, just from a pure empirical point of view, what this data actually looks like and what it actually imply for the types of methods that you want to implement. And then, of course, this is a complex system where you have these nonlinear combinations and interdependence of different factors which affect the dynamics of cholera, but at different levels, for instance, between the human environment level or systems level and the ecosystems level. And then the question that we raise is, what really is the role of rainfall? Does it play a direct and, of course, also an indirect role? Or is it a proxy for something else that is happening also in the rainy season and that we just have not identified? So just to give you a background of the incidence of cholera, if you just look at, this is not about the intensity of cholera, this is just which areas report regularly cholera for the last decade, from 2000 to 2011, using the World Health Organization data. And why I put this up is just in terms of the spread. If you look at the purple of the countries that report most often in that period cholera, and if you look at now at a rainfall map generated where it's just an accumulation of the rainfall over from, this should have actually been August 2011 till July 2012. And you see a similar pattern in terms of where the highest rainfall amounts occur and what you've seen on the previous slide. But what is also interesting on this slide is if you look at everything close to the equator, the difference in the spatial variation over time of where the rainfall amounts vary between the different areas. If you look at the temporal characteristics of rainfall over space, if you were to just extract rainfall data for different latitudes, looking at the latitudinal gradient, you'll see close to the equator bimodal pattern coming through. The further south you move, you get a unimodal, and if you go like around to 20 degrees south, you get not such a clear pattern. Now, what is interesting about this one is, and which is also complicating the development of any prediction model in this area, is that, say, for instance, Uganda is a very small country compared to the other countries in Africa and surface area, but it exhibits, for instance, 3 different types of rainfall patterns, where it has in certain areas just one rainfall season. In other areas, it has 2 seasons with an equal length, not necessarily the same amount of rain if you accumulate it over the period, or 2 seasons with unequal lengths. So we followed an integrative and scale-dependent approach because it became very clear to us that we will have to escalate up and down between temporal and spatial scales to try and understand correlations that we observe and try to find reasons for why these correlations that we observe, why they do occur. And the whole idea of this was to in the end develop a prediction model based on the probability of cholera outbreaks and not necessarily the amount of cases to happen. So you vary between the molecular level right down to up to the global level and trying between these different scales see if you can see any interesting patterns emerging and possible explanations. The area that we focus on, apart from Beira in Mozambique, and that we now currently are working on, is in Uganda, close to the equator. But in a very small part of Uganda, as you can see, is first a map of Africa, then of Uganda and the different districts, and Kasese is in the western part bordering DRC, which also reports a lot of cases, and there's a transboundary carrying of cholera between those 2 countries. And if you now look at the seasonality in the cholera outbreaks, if you just now generalize it just to get on from January to December, you see that the cases do something different to what the rainfall pattern do. You do get the bimodal rainfall season of 4 months on average each. The intensity is not, in terms of total amount, is not the same. But the cholera cases peak in this February-March part after the drier than normal January and December months. And if you look at, we've done a field sampling campaign that is still ongoing, and so we only have data from November 2011 till October. of this here, and you'll see something else also here coming out. We only once detected toxinogenic Vibrio cholera in the natural, in the water samples and sediment samples collected, but there's a large amount of environmental strains in that water, and they seem to peak closer in terms of presence, close in the drier months of the year. So in terms of the results, a short discussion on that, because this is work that we are now busy preparing for preparation. The question of, is it something else in the rainfall season that we have been overlooking and that we should take into account, developing a prediction model based on rainfall? And we know from other work that we've been doing that sunlight, but specifically the UV, the ultraviolet band of the electromagnetic spectrum, plays a role, and where UVA can help repair DNA damage, given that it has the bacteria hasn't been exposed to a too large dosage of UVA or UVB, and but the factors that control the amount of ultraviolet irradiance reaching the surface is actually a couple of factors in the atmosphere control it, but in the that area where we are working, it is cloud fraction that plays the biggest role. And cloud fraction, of course, goes together with the rainfall season. The other factor that came out is that there's a minimum threshold value for the temperature, where most of the occurrence of these bacteria and of the cases occurred is around about 26 degrees Celsius. So we developed a UV index that we adjusted for temperature, and that improved some of our model results. Here's just some more general results. In terms of the normal correlation analysis, using linear techniques of intercorrelation coefficients do not explain really the patterns that we have observed, as you can see on this graph here. And we get much better results when we rather look at similarity analysis and trying to actually relate the characteristics of the spatiotemporal patterns of this time series data to improve our understanding and the results. So in terms of the conclusions in general, is that we need a better understanding of the correlations observed and the interdependency of factors. You have to identify potential cause-effect mechanisms, and you have to parameterize for local conditions and identify drivers or proxies for drivers to allow sufficient time for intervention. From a technical point of view, we need tools to help people to actually do this analysis between these different scales and to revisit what they've been doing so that they can reanalyze and test their results. And this is the people who have funded us and who have been working together with us on this project. Thank you.
All right, we have time for one question. Yes.
You mean like phytoplankton?
This rainfall correlation that you observe, if you have any other measure in addition to rainfall, such as the concentration of what we believe are reservoir for the bacteria in the aquatic environment.
You're asking if we've actually also considered other factors, for instance, something related to phytoplankton or zooplankton that could act as a reservoir for these cholera bacteria. Part of the field sampling is actually includes a very intense phytoplankton analysis. We have not yet established any relationship between that and these lakes. The one lake is on average 4 meters deep. Satellite data is not very helpful in that case, so we cannot make any conclusions regarding the analysis that we've done with the phytoplankton. So to answer you, we are looking at it, but we don't have results to say yes or no.
All right, thank you so much. All right, our next speaker was supposed to be Dr. Rita Caldwell, but unfortunately she couldn't be here, so I will present on her behalf. So what we're trying to do is that we're trying to develop some understanding on how the global outbreak of cholera happens and what are the hydroclimatological controls on the cholera. If you look at this figure, this will show you why we are interested in cholera. If you look at the tropical belt, you will see that this region has the least amount of water and sanitation access. And it has the most prevalence of cholera outbreaks in the region. So about the 50%, the countries which have 50% or less water and access has 70% are more vulnerable to cholera outbreaks in that region. So it's a global disease. In past 30 years, it has been reported in almost all the regions in the tropical world. The red regions here are the countries where you have reported cholera outbreaks. Some of them are endemic regions and some of them are non-endemic regions. So, and these are the two cholera types that are very important to differentiate. So let us first begin with the endemic cholera. These are the cholera regions where you have a continuous persistence of a disease and you see a seasonal outbreak every year. What that means is that you will have a year-round cholera presence, and then you will see peaks in the outbreak during certain time of the year. And this study has been going on for like last 2 or 3 decades. And here the figure that you're seeing is perhaps one of the first studies where authors have tried to characterize the role of sea surface temperature, sea surface height, and chlorophyll. And of course, our understanding has grew far beyond this study. So what we know from endemic cholera is that this is more coastal driven. What that means is that the disease outbreak happens by the intrusion of bacterial waters from coasts to inland. And what happens is that, for example, in Bay of Bengal here, this region has 2 major river networks. One is the Ganges and the Brahmaputra. And during the low flow season, the intrusion of bacterial waters from the coastal regions to the inland regions triggers the first peak. And what happens, and you see a negative correlation between the cholera outbreaks and the river flows. And then you see the positive correlation in the next peak, which is because you have already introduced the bacteria in the system, and then the flooding comes and it accelerates the interaction between Vibrios and the human population. But the challenge is to see what happens in the non-endemic regions where you have sporadic cholera outbreaks, where you, for example, in Haiti, you have 1 cholera outbreak in like last 100 years. How do we account for the hydroclimatological controls on those regions? So what we have, and the biggest challenge is the data, that we don't have enough data to make any generalized conclusion on hydroclimatological controls. So what we did is we collected about 100 years of data from British Indian rule over India from 1875. And here we are going to deal with first 25 years of that data. So here you are seeing 9 regions all across the Indus Valley. And one of the first observations that you see here is that the rivers, all these locations are within 25 miles of the regional rivers. And when I say non-endemic cholera, if you see the right-hand side figure, you will see that cholera happens in like over 312 months. The highest number of cholera months were reported in the Lahore region with a number of like 90 months. And rest everything is like on an average about between 30 to 40. So on a seasonal averages, most of this cholera is often seen to occur between the months of April through September. And what happens here is that we developed a hypothesis to understand how the interaction between hydroclimatology may explain the occurrence of this non-endemic cholera. So here you're seeing 2 rectangles. The red rectangle is the one that is critical here. Here, we hypothesized that if you have air temperature which is above climatological mean for previous 2 months, and which is followed by the above climatological average rainfall, in combination with poor and damaged water and sanitation infrastructure, your probability to have an outbreak of cholera increases dramatically. And if any of the conditions in the red circle is not met, the likelihood of cholera happening reduces drastically. So we checked this hypothesis with data from India and Haiti. So what you're seeing here is the odds of cholera happening when the air temperature is high in previous 2 months. So the black line will tell you here that on an average, the outbreak of cholera, the odds of cholera happening when air temperature is high is about 6 times, 6-folds, which is very high. But alone air temperature is not going to create the conditions for disease outbreak. We need to have a transmission mechanism. But before that, let us look at the thresholds of this temperature. So what we have determined is that the 50% of more cholera outbreaks actually happens when air temperature is greater than 31 degrees centigrade, and this is the air temperature. And what the underlying assumption is that air temperature warms the water temperatures, which then increases the growth of bacteria in the river waters. And now the interesting thing is that if you have warmer temperatures which are followed by above average rainfall in all the regions, you are definitely going to get the cholera outbreaks. And here the black line will show you that the odds of cholera happening with climatologically high rainfall is about 1.5 times. So we think that this hypothesis of air temperature and rainfall is actually providing us with some indication on the controls of cholera in the non-endemic regions. But we are not sure that if this is true. So we moved to Haiti, and Haiti has a recent cholera outbreak in 2010. And what has been presented in literature is that the cholera outbreak started with the soldiers coming from Nepal, and then they're spreading it over to the entire population. And the Mirebalais region is supposed to be one of the origin points of this disease outbreak. So we collected some data from literature, and here you will see that the rainfall is going to play a critical role. And this is the rainfall obtained from TRIM satellite. And so if you look at the previous month before the October cholera started in Haiti, you will see that in last 30 days, there were 26 days with rainfall. And that, in combination with the higher temperatures, which I'm going to show you in a while, plus damaged infrastructure, actually started the outbreak of this disease. And things got worse when you had the Hurricane Thomas, and it lashed the entire country with heavy rainfall, and then it further deteriorated the already minimal water and sanitation infrastructure, and then you see a huge peak in the aftermath of the hurricane. Here you will see the interactions of air temperatures and rainfall. So on your left-hand side, it is the red line is the climatological average, like last 50 years of air temperatures averaged over Haiti, and the purple line is the 2010 air temperature. So you will see that the air temperatures are high in 2010, and if you look at the rainfall patterns, rainfall is huge in the months of September, and this actually led to a massive cholera outbreaks in the months of October. But we were not sure that we are doing it correctly. Then or we wanted to have some idea of putting it into the statistical frame, in a modeling framework. So we developed a modified SIR. This is the susceptible, infected, recover type of model with the active bacterial pool in this, and then we try to calibrate it on the regions in India and applied in Haiti. What we have found is that the outbreak has a very complex mechanisms. We were not able to say that the single person can create an outbreak of cholera in Haiti. What you will see here on your left-hand side is that these are the two scenarios. So each scenario has primary and secondary mechanisms of cholera outbreak. The primary mechanism on your left-hand side is the human-driven, means a single person introduces the bacteria into the water bodies, and then what will happen? What you will see here is that if you have one person infecting the population, you are probably not going to get the epidemic proportions. With 10 people, you're not going to get it. It will take at least like 1,000 or greater than 1,000 people who are infected, who are trying to spread the outbreak, who are infecting other individuals through rivers and through fecal contamination, that will match some kind of seasonality in Haiti. On the other hand, if you have environmentally driven cholera, what you will see is that if you have a threshold-based air temperature and rainfall characteristics, you will see an outbreak which will then match with what we are going to see in Haiti. So this is the work that we are still doing, and we are still trying to understand the intricacies of this model within the context of non-endemic regions. And with this, I would like to focus on the fact that this is a collaborative research that we are doing across different universities. We have active collaboration with Tufts University, and Dr. Shafiq Islam is going to talk about some of the aspects that I have left here. And then, so this is a big collaborative effort that is going on to understand the effects of hydroclimatology on the outbreak of cholera globally. And with this, I would like to thank you all. And one question. Yes, I knew that.
Yeah, I understood correctly. You used your model calibrated on the India data.
Yes.
To reproduce the pattern of you see in Haiti.
Yes.
The results looked quite amazing. I mean, you actually do the same parameters, but you got different patterns for different initial conditions.
Okay, so let me repeat the question. So what you're saying is that I used the same initial conditions and I got the different results?
No. You calibrated the parameters on the Indian data.
Yes.
And then just changing the initial number of infected in the system, you got those different,
Okay.
different results.
Yes. Yes, that's exactly what we did. So we calibrated the model on Indian data, and then we changed the environmental conditions in Haiti, and then we got the results that I have shown you here.
What do you mean by we changed the environmental conditions?
Like for example, the air temperature and the rainfall inputs were different in India and in Haiti. All right, so our next speaker is Dr. Mercedes Pascual, and she will talk about cholera in details as well.
Good afternoon. I cannot see anybody. The light is amazing up here. So I wanted to start by acknowledging first my co-authors, Benjamin Cash, Bobby Reiner, Aaron King, Michael Emch, and in particular our collaborators at the International Center for Diarrheal Disease Research, Bangladesh, Dr. Yunus and Farouk. And the title, this doesn't move forward, maybe.
You click on the right button. There. I mean, left button, I'm sorry.
Good. So this is a more general and alternative title for my talk, a comparative ecoepidemiology of some climate-driven diseases, at least the beginning of it. And in part this is motivated because typical studies on the effect of climate variability have focused on one disease at a time. And in diarrheal diseases, of course, the paradigmatic example has been cholera. We heard a lot about it already, a waterborne disease, a disease of poverty. It has been and is still endemic in large parts of the delta of the Brahmaputra and the Ganges. And this time series from the long-term surveillance program of the ICD-DRB shows you some of the typical dynamics of endemic cholera. It is seasonal, as we heard, and it also has tremendous interannual variation from one year to the next. It is this variability that has led to many studies on the role of climate, in particular the El Niño Southern Oscillation, sea surface temperatures, etc. Now, the disease spreads through fecal-oral contamination of food and water, and also water in the environment. We heard the pathogen survives outside the human host in this environment. And this has led to one view on the pathways that mediate the effect of climate. These would be environmental factors that influence the growth and survival of the bacterium would ultimately influence the incidence of the disease. An alternative view, which is by no means exclusive, would be that, in fact, what varies is the susceptibility of the human host with the breakdown of sanitary conditions, crowding, etc. And in this sense, I think the comparison of different infectious diseases can be illuminated, in particular diseases of the same class that differ, however, in the routes of transmission. And we can ask, for example, whether the environmental effects are systemic for these classes of disease, regardless of particular ecologies. With this in mind, we started to address the dynamics of another diarrheal disease, shigellosis, caused by the genus Shigella, 4 different species, 2 of which, Shigella flexneri, shown here for Madlab, this rural area south of Dhaka, from 1983 forward in the light blue, and Shigella dysentery in red are the ones that cause acute diarrheal illness. And interestingly, they also spread through food, fecal contamination of water, and more person-to-person contact than, at least we believe, for cholera. The infectious dose is much lower than cholera, and that is a difference in the transmission. We will focus our analysis on the cases by Shigella flexneri because it is endemic and occurs every season, as you can see, while dysentery is much more epidemic. And briefly, the outline consists of 2 parts. I will show you some very simple correlation relationships between cholera and shigellosis with ENSO and flooding for both a rural area, Madlab in Bangladesh, and the city of Dhaka, then move quickly to some space-time dynamics of cholera in the urban environment of Dhaka, showing differences between the core of the city and the periphery, and ask whether Shigella shows similar patterns, and with some implications of this comparison. So the seasonality, we have already heard about it. I have it here just for cholera and Shigella flexneri to make the point that we will focus initially on the post-monsoon cases from August to December, when we have the second peak. And perhaps one difference worth pointing out, if I can find a pointer here. Well, no, I guess I'm... Okay, we cannot... Yeah, well, yeah, thank you very much. Yeah, I wanted to make the point that, of course, the patterns, the seasonal patterns differ. In particular, after the increase in the spring, there is no decrease in Shigella, while there is one in cholera, and this may be due to the differential dilution effect in these diseases with very differential doses for the transmission. Now, this is a result we have published a while ago now, but I think it's a striking pattern, and it is here for the comparison with Shigella. This is a correlation map. It is not a map of El Niño. It is essentially mapping the rank correlation between cases in September, October, November, December aggregated, but you can do this monthly with every grid point of an SST data set over the world oceans. And the regions where the correlations are significant are indicated by black, surrounded by the black lines, which correspond to the 90% confidence regions. In particular, you can see in the eastern and central tropical Pacific a region of positive correlation. And this is a little bit better, perhaps. Let me see. Yeah, you can see. Not doing well with pointers. So here it is, this region of positive correlation in the region that warms during El Niños. This is 9 to 11 months before the outbreaks of cholera. Our climate colleagues have then followed this up with an interesting study with a dynamical model that is regionally coupled to the observed SST values in the Pacific, basically to find what would change over Bangladesh for the regional climate following El Niño. And this work basically indicated that the most likely scenario was that after warmer temperatures, we would see enhanced precipitation over Bangladesh. So, of course, the whole idea here was that this would be followed by flooding and then more cholera. Surprisingly, if you do the same operation here for Shigella, you find a very similar pattern. The map in the bottom shows again the fall cases and the spatial rank correlation map showing the same region in the eastern and tropical Pacific. For Shigella flexneri in Dhaka, the data had to be detrended because of this long-term trend that is apparent here. But again, we find similar regions of positive correlation preceding outbreaks with a weaker signal, which I will discuss later. Now, motivated by this, we looked for a flooding index over the country in Bangladesh. These, again, are just very general association patterns. We used basically an index of the percent of land that is flooded for a particular year. But very, very quickly, I just wanted to show that there are outliers, but the idea is that both in the rural area of MATLAB, both Shigella and cholera show these associations with flooding, and similar patterns for Dhaka, with much more noise here for Shigella in the bottom right. Now, of course, I said that I would discuss these weaker associations. We are aggregating the cases of a very large urban environment, which can be problematic. And to address whether this is perhaps one of the reasons we see these weaker associations but similar patterns, let me summarize some recent results we have published, showing that in fact the city is structured into regions that appear to behave differently. Maybe I'll manage to do this. Is it possible to put the arrow here? Yes. This is a very bad idea, but sometimes it works. So we basically took the data, and I just want you to look at what happens during these peaks. At the bottom, you see the aggregated El Tor cases. When an epidemic happens, you see that, in particular, the core of the city with the small sub-districts lights up. And this is a very stochastic dynamics of the cases, with extinctions in different parts of the city, and essentially the appearance of cases in other parts. These are sub-districts of the city known as tanas in the city. And the historical center runs along a river and is shown by the smaller districts. You can see again when the cases start climbing up that there is a suggestion, if you look at this movie for longer than you are interested in, you would perhaps think that the center behaves differently than the periphery. So, if we actually wanted to test this and developed a model for the disease, so oh no, not the movie again. You don't want to see it again. Okay, thank you. So then what we did was to take the data for cholera. We did not develop an epidemiological SIR model as the ones you saw before because of the very stochastic nature of the disease, many zeros. These are just the reported cases. So we decided to basically classify the disease into low cholera, high cholera, no cholera, just these discrete states, and work with a model that had probabilities of transitions between these states from 1 month to the next, and then these probabilities could be modified by both the season, the state of the neighbors, climate covariates, and so on, flooding, and also importantly, which part of the city you are in. And we consider here whether we had to separate 2 groups in the city or just 1. And the results of this work, which I think are most relevant for today, are that there is a very significant role of spatial heterogeneity in the sense that the 2 groups of districts were needed to capture the dynamics. Otherwise, it was impossible to fit them all. In fact, you see a very significant p-value for this distinction. Interestingly, there was also an interaction between climate forcing and spatial heterogeneity. That is, the effect of ENSO was significantly different for the core of the city and the periphery. In the figure you see down there, we tried to map some socioeconomic factors, but that shows some relationship to this distribution, these 2 areas of the city. In particular, here in the left, this is the population density with, this is 100,000. There is no mistake in the zeros. This is 100,000 per kilometer square in the center of the city. And this is the percentage of the poorest type of housing. So there is a river going through here. What we do not have is also information on how flooding is distributed at this spatial resolution. But that would be extremely interesting. So what about Shigella? I will not show you the movie. I wanted to make the point, just 2 main results on Shigella. We have now the data down to this TANA level. And here I'm showing you the TANA sorted from lower, yes, I'm getting done. Sorted from lower to higher. The green ones are districts we had thrown away in cholera because they had no cholera, periphery one. These are the next in the periphery, and this is the center of the city. So there is this clear distinction, and if you actually run this probabilistic model for Shigella, you also find a highly significant effect of spatial heterogeneity for the same regions of the city. So in conclusion, the association we see with ENSO and flooding is apparent for both diseases, and there are similar patterns of spatial heterogeneity in a large urban environment for these 2 diseases. I think this underscores systemic mechanisms most likely related to human susceptibility with the disruption of sanitary conditions and access to water following floods. And this raises the question of the environmental persistence of Shigella flexneri, perhaps, but also I think the question of working with higher resolution patterns of flooding within an urban environment, which would be very interesting. Thank you very much.
I don't think so. We have luxury of asking questions to Dr. Pasco.
Oh, no.
So we have to move to our next speaker, and our next speaker is Jody Stryker, and she will talk about malaria.
All right.
Forward and backwards is the easiest way.
Okay. Okay, hi. My talk today is on a study that looks at the impacts of land use change on mosquito population dynamics in a highland region of Ethiopia. So if you guys have been here for the whole session, you actually saw this figure before, but I'm still going to touch on a couple things because I think it's important for everybody to be on the same page. Malaria is a vector-borne disease that still affects many regions of the world and is still very difficult to control, largely because of the interdependencies among the parasite that causes malaria, the mosquito vector that carries the parasite, and the human host, who is critical for continued transmission of the disease. Another thing I want to point out about the cycle that's relevant here is that the development of the mosquito from egg to adult, so this portion of the life cycle here, thank you, is highly dependent on a number of factors, one of those being ambient temperature, another being the persistence of suitable development habitat in the form of shallow pools. Due to certain limiting factors on this life cycle, certain areas experience only seasonal or epidemic malaria as opposed to endemic malaria all year round. Populations in these regions are particularly sensitive due to low levels of immunity to the disease. Also in the last 20 to 30 years, regions such as these have experienced, including highland areas of Ethiopia, have experienced several severe malaria epidemics which have caused particularly high rates of mortality. So with seasonal and epidemic malaria, transmission is often restricted by elevation and the corresponding lower temperatures. But in highland regions where malaria occurs only during the warmer months, it's also limited by seasonal rainfall, which under the right conditions provides enough water to form pools on the ground surface, thereby establishing the primary breeding habitat of malaria-transmitting mosquitoes. And some of those, these are some of the pools from our field site in Ethiopia. Increasing population pressures and land use changes in these regions, such as the conversion of agriculture from local crops like teff to maize, have an effect on hydrological mechanisms such as runoff generation, possibly by increasing surface water and resulting in persistence of malaria, higher persistence of malaria in these regions. So this research aimed to investigate the physical mechanisms linking overland flow and malaria transmission. And specifically, we isolated the effects of land use alone on runoff generated and thereby on mosquito abundance using a coupled modeling approach. So this modeling approach was applied to parts of a village called Waktola in a central highland region of Ethiopia. Elevation there ranged from about 1740 to 1800 meters above sea level, which puts it right at the fringe of where malaria transmission is possible due to those lower temperatures. We used for the modeling study, we used a 2 kilometer by 2 kilometer area near the center of the town where land was used largely for agriculture as well as pasture. The model domain encompassed several of these shallow depressions where water regularly pooled and formed successful breeding habitats. These recurring pools were monitored for larvae at least weekly through 2 field seasons, and light traps were used to monitor adult mosquito abundance near those pools. And as I touched on earlier, agriculture is increasing in this region, especially the farming of maize. And at the same time, it's been observed that seasonal malaria is also increasing. So we're really trying to get at some of the specific hydrological mechanisms that are affecting these changes. So we also used a model called Hydromats, which you guys also heard about a little bit earlier. It was developed at MIT and is comprised of a physical-based distributed hydrology model coupled with an agent-based entomology model. So here you see a couple of the GIS-derived inputs to the hydrology model, including a high-resolution DEM, which was obtained from the 3-arc-second product from Shuttle Radar Topography Mission, as well as soil and land use maps derived from field observations as well as digitized Worldview 2 images. The DEM was interpolated to a 10-meter resolution and was also manually modified where necessary to more accurately represent those shallow depressions that we saw and that were observed to be breeding habitats. In addition, a meteorological station was also installed at site that measured precipitation, temperature, net radiation, humidity, as well as wind speed and direction. So that data was measured at the site and inputted into the hydrology model as hourly averages. Also, you see on here, we measured soil moisture at several locations as well as water depth of a few of the pools. So you see those on the map as well as the location of some of those recurring pools and the light trap installations. So the hydrology model solves numerically the overland flow equations to calculate surface water flow as a function of water in excess of soil infiltration capacity. And then it outputs surface water presence and depth at every time step. So this is model output for an arbitrary time step, just to show you guys. And you can see that these pools can form either as, as a single cell or as cells together, where these pools can vary with depth also, vary in depth. The location and depth of these pooled water at hourly intervals, as well as the meteorological data for the site, become the inputs into the entomology model. And based on the predicted length of time these pools last, as well as the specific depth and temperature of the pools, the model calculates the number of mosquitoes that develop in and emerge from each pool location, as well as tracks the total number of mosquitoes present in the environment at every time step. So once the mosquitoes emerge as adults, each mosquito moves probabilistically within the model domain, where flight can be affected by wind as well as the location of carbon dioxide emitting hosts such as humans. Here we considered humans stationary based on the assumption that mosquitoes feed mostly at night when people are in their homes. So you can see these, the green spaces are basically our representation of human locations in the model. And one of the important aspects I want to point out of this model is that it does represent the spatial co-occurrence of mosquitoes and human locations, as well as the locations of those breeding pools. The agent-based model also tracks a set of characteristics for each adult mosquito, including its X and Y location in the model domain, and a number of other characteristics, including time since last blood meal and infected status. So here you see all the blue dots are all of the mosquitoes that the model thinks are present in the model domain at this time step. And these are the ones that are currently infected with the malaria parasite. So as I mentioned, we measured soil moisture using time domain reflectometry probes at 4 depths at a total of 5 locations on site. And the hydrology model was calibrated to 2009 field conditions, and 2010 runs were used as validation. So this is volumetric water content measured and modeled at a depth of 5 centimeters for 2 different locations on site. The major parameters calibrated were surface roughness coefficient, total leaf area index, and a root zone parameter, all which were assigned based on land use. Mayonnaise roughness coefficient represents resistance to flow by vegetation present on the ground surface. Leaf area index is the dominant parameter affecting evapotranspiration, and root zone parameterization affects water uptake by plants that are present in that grid cell. So we saw a similarly good fit at most of the monitored locations, particularly at shallower depths, indicating that the model was doing a relatively good job of representing soil moisture conditions at the site, specifically near the surface. And the near surface conditions are the ones that are most important here, because those are the ones that have the dominant effect on partitioning of water from between rainfall and infiltration, or between runoff and infiltration. So the entomology model was last implemented for a relatively similar environment and for the same species of vector mosquitoes. So the only parameters are adjusted here were effective flight velocity, which has an effect on adult mosquito flight, and ecological carrying capacity, which limits breeding in pools and depends largely on nutrient availability. So here the cumulative number of mosquitoes as measured by the CDC light traps through the wet season and up until October 15th was about 30% less in 2010 than in 2009. The cumulative abundance predicted by the model for the 2 seasons, also up till the same date, showed an approximately 40% decrease. So I want to note, I realize that the numbers of mosquitoes that you're seeing here look extremely different, and a large part of that is due to the high degree of error associated with the field measurements of mosquito abundance. But what I really want to point out is that the model can capture relative differences in population abundance due to variability in seasonal rainfall and temperature and other climate drivers. So here now are the cumulative number of predicted mosquitoes, also at the end of the wet season, for this time for each of the land use scenarios that were tested. All of these were run with 2010 meteorological data and ultimately compared to the 2010 baseline scenario, which is there on the right. Again here, the number of mosquitoes is really not what's important. What I want to highlight is that in general, the land use types characterized by, or the land use scenarios characterized by increases in agricultural land usage, especially increases in row crops like corn, produce an increase in mosquito abundance by the end of the wet season. So these land usages resulted in increases in surface runoff and higher persistence of breeding habitats in those runs. And in contrast, land use types that presented more or represented more area as rainforests or trees typically resulted in a decrease in a mosquito abundance. So in those runs, pools were more likely to dry out, breaking the life cycle of all mosquitoes in that pool. So results of the study indicated that land use type, as well as the spatial relations between land use, change, and breeding habitats, are important influences on the persistence of these breeding pools. The ability of models to simulate small-scale hydrologic responses to land use changes demonstrates their usefulness as a tool for understanding and possibly for predicting interannual variability in mosquito vector populations, as well as for looking at their responses to outside influences such as population growth and movement, as well as changes in precipitation patterns and variability. So results of the study also add to previous work linking land use with microclimate variables such as temperature and humidity, which in turn influence the development of both the mosquito and the parasite, thereby shaping local mosquito population dynamics and variability in malaria transmission. We recognize that a number of factors contribute to variability in mosquito abundance. Some of them, such as temperature, have been studied extensively, and others, such as land use change, are less known. So this study was really one of the first that uses numerical models to mechanistically simulate the link between hydrology and variability in intraseasonal mosquito abundance, and shows that land use has an important influence on both the formation and the persistence of these breeding pools. And suffice it to leave that up.
All right, we have time for one question. Quick one, though.
I can't see anything.
Okay. All right, we have two. I'm divided. Yes.
Can you speculate about trends in land use and trends in malaria? You mentioned that there have been an increase in malaria cases.
Yeah, I mean, in general, I actually. No, that's okay. There has been, there was one study that I looked at that shows, like, if you look at warda health cases in specific wardas in specific regions, you can see that there's an increasing number of malaria cases just during those wet seasons in areas very near to where we did this study. So that was part of, but then in general also, like I said, in the last 20 or 30 years, there's been sort of an escalating number of these large epidemics in regions that are right on the cusp of where transmission is typically either epidemic or seasonal or even non-existent. So, and the increase in maize is that one's an obvious one. So that's sort of where we got the idea for looking at that effect.
All right. Okay. Thank you. Our next speaker is Andreas Kolodis. Did I pronounce it right? Perfect.
I might need to see slightly. I know how it works. Thank you. Yeah. Good afternoon, ladies and gentlemen. I would like to present this work on behalf of my colleague David Rickerby, who could not travel to United States for this presentation. It is collaboration work.
Left side to go forward.
On the Ear to Heaven project, a project that has been financed in the 7th Framework Project program of the European Commission. And this is primarily related with remote, this project has been related with cholera by combining remote sensing and in situ observations. But the part that is related to leptospirosis is a part that has been initiated in a collaboration between World Health Organization and the Joint Research Center, which is an institution of the European Commission for Research on Environmental Sciences. A few words about leptospirosis. Leptospirosis originates from zoonotic pathogens associated with asymptotic rodent carriers, affects humans via various different and direct and indirect routes, and claims many victims with large outbreaks during climate-related disasters. I would like to show you this map of the relevance of diseases produced by WHO. And as you can see, the incidence of leptospirosis is here, very much related to the flu, measles, malaria, and yellow fever. So obviously this is one of the diseases that you might have not heard of recently, but you are going to hear about it in the future quite a lot. WHO indicates that these incidents are highest among countries with high frequent flooding, and it affects primarily individuals and geographic regions with limited resources, which are subject to higher rates. My presentation has 4 parts. The 4 parts is primarily the motivation, the regulatory element, which involves WHO and the European Commission, of course, a technological foresight part, and perspectives and remarks, which terminates my presentation. Motivation. The motivation is primarily these 5 bullet points, where we have information technologies which can lead to radical changes in the way we perceive and understand and manage water-related severe effects. It is necessary to have real-time monitoring of pollutants and primarily pathogens in this particular disease. Data must be of sufficient quality, and this is why we have to integrate remote sensing and in situ measurements. We have to have current research in enabling solutions primarily for items that we have not detected until now. Primarily the diagnosis of the leptospirosis is a very complicated and very expensive disease mechanism. And finally, we have to have early warnings and verify compliance when we take measurements, especially since this disease can be confused with other type of diseases like dengue fever, etc. On the second part of the presentation, I would like to say a few things about the regulatory aspect, and here I dare to put in front of you some of the regulations that they are associated with the Water Framework Directive in Europe, primarily not to introduce you about regulations that may have very little relevance to you, but primarily in order to show what we need to do about preventing further deterioration, that we need to take, promote sustainable use of water. We need to have a reduction of discharges, and finally we need to prevent further pollution of free water resources as we know them today. And out of this Water Framework Directive, I need to tell you that we have the following requirements that they are here. Requirements for analytical methods and monitoring tools that take into account endocrine disruptors, phenols, atrazines, pesticides, pharmaceuticals, dyes, and sulfonates. And we need to do that simultaneously without sending samples in the laboratory. And we have to have, if possible, a multi-residue analysis of water pollutants on the basis of these things. Coming to outbreaks of leptospirosis, the problems that we have about this disease are, of course, first the management. And on the management aspect, we don't have, and we need to have diagnostic tools that they are not currently to the level that we would like from the clinical aspect. We have to take into account that delayed in health treatment is particularly critical. So early detection and treatment is something that is necessary for the management of the disease. Concerning the prevention of the disease, we need to have to improve the surveillance of animal species involved in reservoirs. We have to take and deal with underlying environmental problems that, driving the disease transmission, and such type of environmental problems is the floods, together with population and livestock, affecting not only population but also livestock, because that might cause problems afterwards, and also take into consideration the poor infrastructure. Fortunately, the poor infrastructure does not change very severely, but require remote sensing images that they are very high resolution and take into account the population density on the time that a severe event is going to take an effect. About leptospirosis outbreak, the finite element that we need to consider of is primarily the prediction, and here we have to have into prediction means of course models, and we need to take into consideration models that they are predicting the acute events for endemic annual events, and we need to have models for the infection dynamics, especially for rodent reservoir population. So obviously the models concerning the environmental extreme events are macro scale for water cycles, typhoons, cyclones, and or hurricane formation on an annual basis. But also on the micro scale, it is very, very important. We have to have into consideration the onset of flooding and the length of time standing water is staying there and influencing both rodent and human population. Concerning the infection dynamics, we have to take to answer questions like the threshold of the disease, especially in particular with rodent population, and we need to know white densities below which transmission is low and has very low probabilities for the causing of outbreaks of leptospirosis. Very quickly on the importance of the high spatial resolution, here you see that we have a specific need to couple grids of different resolutions until we come to the level that we need to do, and we have to do them with real-time coupling. That means that the modeling in the inner grid must influence the outer grid and vice versa. Entering into the third part of my presentation, I would like to say that out of this discussion, it is quite essential to focus in this particular moment on developing sensors for monitoring and water and wastewater quality. This will improve measurements inside and will lead into a completely decentralized controls in areas where they need to be taken into consideration without the need to send samples back to laboratories for expensive verification as it is done today. A few words about my title has the remote sensing and in situ. So I dare to take this extract from Science, where if you see the forecast of what will happen between modeling and remote sensing and in situ, you will see that in situ sensors are very, very much in the bottom line. There are very little progress that has to be made, and it needs to be changed in the coming years if we need to affront diseases that we have not been able to affront until now. In situ sensors, they have been suffering until very recently about accuracy, selectivity, calibration, reliability. There is a need to have manufacturing in nano scale. They need to incorporate data architectures for which we need to have a sophisticated means of citizens' participation and citizens' sensor devices. And also we need to have a telematic cover in areas where we need to incorporate such type of in situ technologies. Particularly for leptospirosis, I'm showing you here a sensor device that has been developed in the 5th European Framework project. It is costing in the region of 3,000 to 5,000 euros. Simultaneously, it injects here a laser beam, and the laser beam in this particular plate, according to the color that you see here, is simultaneous analysis of several types of molecular imprinted polymers, but also can be converted to take into account pathogens. The beauty of this type of devices is that it takes 18 minutes to analyze 32 type of different parameters, and it's capable of having 500 measurements before regenerating this type of sensor device. Here is the device as has been developed. It takes into consideration an autosampler that automatically takes water samples and feeds them into the system here for this analysis of the AWACS system. There is no need, because I see my time here is progressing, so I need to have here multi-analyte measurements, especially for inlet calibration, has shown that the limit of detection is in the level of pre-PQ, and in particular this type of sensor devices have no pre-treatment or pre-concentrations are needed for calibrating this type of devices. So these devices are stable and they can be utilized in site without very high expertise of laboratory experience. Concerning the other element that needs to affront such type of in situ sensor devices, of course it's a network architecture where it involves the involvement of citizens, that they will be in a position to have sensors in telematic devices, multi-sensor capabilities with telematic devices, that they utilize the standard Wi-Fi protocols that transmit the data in a central service where they can take a control in a remote way. A little bit further ahead, and this is the 7th framework program, we are considering that such type of sensors could be utilized in underwater robots, and we have such an experiment which is associated with primarily measuring salinity, temperature, underground temperature, or other type of parameters for simple environmental monitoring, but they can be incorporated to detect pathogens if we need to take into consideration such type of things. Take into consideration that for the time being, in the frame of the GEOS project, there are approximately 3,000 buoys that they are deployed at since for conventional environmental data. So we have an abundance of environmental data that can be used for the sake of environmental-related diseases. Lepto outbreaks, this is primarily my last slide. I have mentioned to you that for leptospirosis outbreaks, we need to be concerned about management, prevention, prediction. Here I am underlining where in situ devices they can be actually taken care of. Here we have a combination of in situ and Earth observation. Here we have only Earth observation. Here we have in situ technologies and constellation of Earth observation devices. For those of you that have followed the NASA, but as well as other conferences like the Pleiades deployment of satellites, we are talking now not about a single satellite, not about a single instrument, but we need to talk about a constellation of satellites that they will be in a position to provide important data in areas where we need to make our presentation. This is my last minute, and I would like only to show simply the GLEAN initiative, which is the GLEAN initiative for Global Leptospirosis Environmental Action Network. And here is the incidence in the various parts of the world. And my final remarks in a very, very quick way. Biosensors can be integrated with telecommunication and Earth observation in order to detect several types of environmental contaminants and toxins, and it can produce multi-analyte analysis with capabilities for simultaneous measurements. It facilitates remote surveillance and control, and it has an improved spatial resolution, and of course it is very simplified and reduced costs, but complicated due to the interdisciplinary information needs that they need to be merged for solving this problem. Thank you very much for your attention.
I think we have to move to next speaker. Thank you very much. Our next speaker is Dr. Shafiqul Islam from Tufts, and he will talk about cholera. Oh, it's this one.
Good afternoon, and thank you very much really for giving me the opportunity to have the last word. Not really. I think I don't know why I was scheduled at the end, but I think it gave me an opportunity to listen really what I was going to say, and I also changed some of these things as I was listening. But I could not change the slides, so I'll just try to improvise this by just speaking. As you can see, really, I did not even put a name because this is really not an individual effort. This is a very large team effort, and from all kinds of institutions here in the US as well as in Bangladesh, from Tufts to Maryland to Virginia and Florida, Johns Hopkins, ICDDRB, and many more. So what we are trying to do really here, and I'm very pleased to see this really, we're trying to create some type of really mechanism so that we can bring water and health together. And we're coining this term called hydroepidemiology. Whether it will stick or not, that's not the point. I think what we have heard here so far is that there is a very strong connection between water and health. And we knew this, and we knew, as you'll see really, we knew this for quite some time. But just knowing something and doing is not the same thing. So to give you an example, this is a data set that is about cholera in Bangladesh. If you look at the prevalence really is the blue lines, and that has gone up. On the other hand, the case fatality has gone down. So this is a remarkable success. And if you look at ICDDRB, they have been nominated for Nobel Prize for many times, and hopefully they'll get one. Because they were the one, they have created this significant reduction in mortality in cholera in Bangladesh. That is a model really all over the world right now. If you look at the mortality is almost 0.1% in Bangladesh. But the prevalence has gone up. So that means we have not been able to contain the disease. The question is then why? That is, I think, is the question we need to ask really. It's not that basically we could not really reduce the burden at least when it hits us. But it is still hitting us and creating significant amount of disease burden. The second one is really we also sometimes get very confused. Confused with signals and the noise and the like. This is a paper we published really from our group last year talking about warming oceans, phytoplankton, and river discharge. And as Antar was showing in his slide, really, there was a paper that was published in the 90s where trying to relate sea surface temperature with cholera, with a very strong correlation. And that paper was cited many, many times to try to make a causal connection between sea surface temperature and cholera. And what we found really there is some interesting contradiction there because when sea surface temperature goes up, really phytoplankton goes down. Usual understanding is that if phytoplankton goes down, cholera should go down, but cholera goes up. So there has to be something else going on there. And what was going on in the Bay of Bengal really is no sea surface temperature, because it also has very large rivers coming in, and it brings in a lot of nutrients. That nutrients really changes the ocean dynamics. So that is not the causal connection with cholera and sea surface temperature. When this was published, really, something else happened. It was picked up by a lot of people, including the New York Times, and they made the connection that climate change is not really a culprit in increasing outbreaks. That's not what we said. We did not really make any connection with climate change. Really, this is a very difficult question. We don't want to even go there. What we said really that if there is a climate change, at least general consensus is that some of the things will get extreme. Drought will get extreme, flood will also get extreme. If that happens, then cholera will get extreme in both cases, because there is an asymmetry between those two. And then someone else basically got into this, and he is the Joe Rom. He is considered the most fiercest blogger of climate in New York Times. And he said cholera and climate, New York Times gets its story exactly backwards. And this is exactly what is going on. We often really get confused really with the signal and the noise. Unless we are clear really what is the signal and what is the noise and what we are trying to do really, this problem cannot be solved. And that is where I think the science and the AGU can be very powerful. And this is not new really. This is a book I found really almost like 300 years old. So at least about 300 years ago, maybe even longer, I think, they have figured out that there is something going on with the weather and the disease. And it took us that long now to understand that, yeah, climate change will have an impact on weather and will have impact on health. We knew it for 300 years. And then, of course, snow came and we were looking for cholera everywhere and could not find it. And now, of course, we bring in satellites, see whether can we find it from the satellites. None of this really will give us, unless we can find out really what the connections are. And maybe causal connections are not the right thing to look. Perhaps we have to look at the enabling conditions. The example that, for example, the Antar was making his presentation about how to identify non-endemic cholera, I think is quite interesting. Is that can you really look at really certain threshold of temperature and certain threshold of rainfall and then make a connection? That connection may not be differential equations. That connection could be heuristics. And same thing there basically was Dr. Pascual was trying to show shigellosis and cholera can also have some interesting connection with water. And so how is it? It may not be mechanistic. It may depend on certain thresholds. It may depend on certain particular conditions that exist. The question and the challenge is to identify those conditions. Can we identify those particular conditions that lead to particular outcome? Unless we can do this really, this hope really, we should be able to do it from first principles, I think is maybe a little bit misplaced. The reason it is misplaced really, what we are trying to do, we are trying to look for clean problems. This is not a clean problem. This is a very messy problem. The messy problem does not have clean solutions. So we first have to admit this. Once we admit this, then we can look for solutions really. So what I call this really, this is the mess that we have. So I'm saying that there is a micro level mess and there is a macro level mess. And we are basically trying to find out how to do this. And macro level is the hydrology, climate, and so on. Micro is the one you go actually into the genes and try to find out. And these two groups are not really talking to each other very well. Right now, you can basically have a very good vaccine for cholera. We are talking about really, yes, I will try to relate with climate and so and cholera. But unless we somehow try to combine these two, this problem cannot be very easily at least addressed. I'm not saying that it can be solved that easily. So to do this, really, we make a significant distinction. There is a micro and macro. And since there are new biotypes every day emerging, there is simply no way really we're going to get rid of cholera. So it will be with us. So given that, then what can I do? So the thing to do then is to be able to make some forecasting. Can we do this really, and how quickly can we do this? So I borrow basically a picture also now from malaria. So malaria, so you have a micro level mosquitoes, and then you have macro level signals. And is it possible to connect these two? If it is really, does it really allow us to make some forecasting? That's where the basically challenge can come in, and that's where the hydrology and the water people can contribute. So if you look at this 7 pandemic, what we are seeing that the first cholera really starts from the coastal regions. And if it does, if you go to Bangladesh now, you see a very significant difference between two types of cholera. In the spring, you look at the left-hand side, is the drought-prone region where you get the first cholera hit. You never get really the cholera in Chatok, in Sylhet really, first one, in spring. Why does it not happen really? It happens when it floods. So that means there is an asymmetry. So I cannot have a simple model really that will explain this. So I need two different types of models, one model for drought, one model for flood, and recognize that there is an asymmetry there. Once I recognize that this is what I'm calling this enabling conditions. The model that is good for drought is not going to be working for the flood. So you need two different types of models. If you can combine these two, then you have it. So that's what we did really. We did this, and we have several papers really to show really how this asymmetry can help us really in developing forecasting models. And you see this almost like a uncanny, really. If you look at the left-hand side, the salinity is very high. At that time, you're very close to the coastal region, and you get a very high cholera in the spring. On the other hand, when the flood is high, the stream flow is very large, you get the second peak. So this relationship really is not linear. It's asymmetric. The question then is, we need to build a model that can exploit this. And same thing, now from the satellite, I can look at all this phytoplankton, and that can help me really with the spring cholera in Bangladesh. And then I go to the flood, fall peak in Bangladesh. So both can be done from satellites, but looking at two different types of variables. Now I come back really from malaria. So this is another example I borrowed from my friend at MIT, really from Al-Tahir's group. So they're doing some very interesting work. We're trying to relate hydrology again with climate. So can I really detect some of these large-scale signals? So this is the signal for malaria. Now if you look at rainfall, very interesting, there is a lag. So if I can predict now rainfall, I should be able to predict malaria. That is the type of connections we're looking for when we talk about hydrology, climate, and the connection. And this type of connection can be detected, can be forecasted so that I can make preparation to do what? To reduce those prevalence rate as well as mortality rate, not only one or the other. So this is basically a big mess that we are in right now. So we don't know exactly how to get through this. This is our conceptual view map you can think of really that we are trying to figure out. Given that I know sea surface temperature is high or phytoplankton is high in the Bay of Bengal, can I really make predictions of cholera in Bangladesh, in Dhaka? How far can I do this? Can I do it 2 to 3 months in advance? That's all we're hoping for. I do not want to do it for 1 year. Even 3 months lead prediction, if you can do this, that can significantly help in terms of reducing the not only mortality as well as the disease burden, because we can let the people at ICDD get prepared. You can think of a really very dire situation. If there are 10,000 people really get sick in Dhaka, it is almost impossible for even the hospitals to take care of that many people. So if I can make that predictions really at least 3 months ahead, they can get prepared. And some of the years it will happen. When if you have a very significant drought, the likelihood of that having very high cholera in this spring as well as in the fall in Dhaka is also very high. If that likelihood can be made now at 80 to 90% rather than 50%. 50% is flipping a coin, doesn't help. So I have to basically make it better than flipping a coin. The question then is, can hydrology help me really to make that prediction? And perhaps it can. And we have done some simulations really to show really, if I look at different regions where there are double peaks and single peaks, if you look at the southwest region, it doesn't have double peak. Although people say that there is a double peak in Bangladesh, no, double peak is only in Matlab and Dhaka. Double peak is not in Chatok. Double peak is not in Matbaria. Because the drivers are different. In Dhaka, both drivers are important. In Chatok, only one driver is important. So we need to understand really where the particular driver is important, then only we can make predictions. And we have done some preliminary calculation to show really it can be done. At least for Dhaka and Matlab, it can be done fairly well using part of the data set for calibration and part of the data set for validation. The key now is that what are the key dominant variables I need so that I should be able to make this model. These are localized. So these are not general model that I can take this model. Even in Bangladesh, I cannot apply it into two different regions. That is what the challenge is. It is really, if our hope is to create Newton's law, I think we should be careful. It simply cannot be done. So I'm just making a very bold statement here that we are not looking for Newton's law. What we are looking for really is contextual models. Those are based on mechanistic understanding of the problem and the variable that can be easily measured. If we can do this really, then I think the possibilities are tremendous. So I have just listed few of these diseases, those are related to water, and in many different ways. So the question and the challenge for the community as a whole for us is that now how can I take each of these diseases and try to relate it to macro scale variability, not micro. So macro scale variability will give me a longer forecasting time. Can I do this for cholera? Can I do this for malaria? Can I do this for typhoid? These are the type of questions we need to ask. With the understanding that this is what we are trying to do. We are trying to essentially minimize the prevalence also, not only mortality. That is only part of the game. Prevalence itself also creates significant burden really for the system itself. So with that, I'll stop. Thank you.
We have time for one very short question. Please.
So the observation of the predictions of your simulations that you just showed us from 98, were those from initial conditions? So are you showing the simulation for those 4 years in a row?
So these are not simulations really. Okay, so I think the question is that basically, is this really a simulation or is this a mechanistic model?
No, the question was how far, because it looked like you could simulate and predict for 4 years. How far, or are you just doing 1 month ahead, 2 months ahead? So I'm sort of asking how far into the future were you simulating your predictions?
All right. So the model that we showed really here, we are only showing that this model can essentially create those simulations. This is different than forecasting. Forecasting model I have not shown you. Forecasting model, we have a different model where we are using, for example, only 2 variables. Only the variables from the phytoplankton, sea surface temperature, and in 1 or 2 cases with the air temperature. But this is a paper that is in review where we are doing 2 months ahead predictions. So using, for example, I can tell you really if I are in September, I'll tell you what will happen in November. So that is a different model where it will be actually operational. Here what we're trying to see really, can we really find out the driver, those are mechanistically derived, to use see really what this model works. So can this do this? So if I calibrate this model like with salinity, with flood extent, and with other couple of variables, is it possible to at least follow the trajectory of the cholera incidences? That's what was shown there.
You're following the trajectory completely from environmental drivers?
That is correct, exactly. So basically, it is basically dominated only by the environmental drivers. We're not using anything. So we don't know really what the first outbreak is. We're not going into the micro level. It is purely driven by macro level drivers. That is exactly correct. And I think that is an important distinction because that is exactly what we want to do. We want to be able to use the variables, those are easily measurable and perhaps predictable. So if, for example, sea surface temperature is predicted by, say, NOAA 6 months ahead, I can use this prediction to put into my model. So then I have a 6 months lead time. So that will then depend on really what is the prediction lead time of the current understanding of that variable by the community. If I want to use flood extent, then I need to rely on someone else because I am not basically good at really predicting sea surface temperature. And I don't want to do that. I want someone else to do it. So maybe that's a misnomer. So I just want to take one quick minute really. So we have something else going on tomorrow. So this is a pitch I am making. So there is a session at 1:40. It's a union level session that we are organizing. We're calling this Unsolved and Emerging Problems in Water. So the idea really here is to engage the community to pose some difficult questions in water. So difficult meaning this, so the motivation really came from Hilbert. In 1900, he posed 27 problems in mathematics. And that has dominated mathematics for the last 100 years. And some of the problems are still unsolved. So we are not really there for solutions. We are hoping that at least AGU gives us a forum where the water community can start posing those questions. So hopefully at least some of you will show up there and maybe pose some questions really in this interface between water and health. What are some of the difficult questions? So this is the premise that we want to ask the question, how does it help science and then serve the society? That's the first part of the question. Second, why is this a hard problem? And what do I need to do now so that I can start addressing this problem for tomorrow? Thank you very much.
Thank you, Dr. Islam. All right. Thank you so much for being here, and I thank all the speakers for keeping their presentations on time. And with this, I would like to end this session. And thank you so much.